As you can see, I'm going to be presenting on the gut microbiome of lions in Etosha National Park. So just some background before we get into it. The gut microbiome is strictly defined as being made up of the bacteria, archaea, fungi, and viruses that live in the digestive tract of animals, as I'm sure most of you know. Now, most of what we know about the gut microbiome comes from research on humans, which I think Louisa will be presenting on after this. And animals of economic significance like livestock and so on. And what we know from these studies is that the gut microbiome has quite a significant relationship to health factors in the host and specifically the immunity of the host and a variety of other phenotypic factors. What we know relatively less about is the gut microbiome and its relationship to the host in wild animals. What we do know from a few studies is that the gut microbiome of animals in captivity is quite different from those in the wild. And so what you can see here is some results from a study that showed that the diversity of the gut microbiome in captive animals is quite significantly lower than their wild counterparts. And there's a variety of reasons that could be contributing to this, some of them summarized on the side here. Specifically, one of these wild animals is, of course, the lion. Now, there's only been one study done on the gut microbiome of lions, and this was a study done in Asia on a subspecies of lion, the Asiatic lion, done on 3 lions rescued from a circus in India. And what you can see here are some of the bacterial genera present in those samples. I'd like to point your attention to the dark blue abundances, which is the genus Fusobacterium, which was found to be most abundant in these Asiatic lions. And so considering this, we decided to focus my research on the lion gut microbiome because of the small sample size of that study and the lack of research on the African lion. And so the objectives of our project were these 4, as you can see: to characterize the core microbiome of the lion gut, to perform functional analysis on the bugs in the lion gut, to calculate alpha and beta diversity, and 4, we wanted to see if we could identify any variance-causing factors. But for the purposes of this talk, I'll just be focusing on the first objective, just to characterize the lion gut microbiome. And so in order to do this, I went to Etosha National Park in the north of Namibia with a group of researchers, and we collected 23 samples from 20 lions. And I extracted the DNA, and we sent the DNA for both shotgun and long read sequencing, thanks to Ansia and Nadia at SAMRC for the long read sequencing. And so first we'll be going through the short read sequencing, which is mostly complete. You can see the data analysis pipeline here with the pre-processing in green, and then we split that up into the classification of the short reads in blue and assembly and binning or generation of metagenomic assembled genomes or MAGs in orange. And so to start, we'll be going through the blue section, and I'll just quickly take you through our genome taxonomy database or GTDB classification of our reads. So what you can see here is our lions on the left side, and then we took the 3 reference lions, the Asiatic lions, and also classified those through our pipeline. And what I'd like to point out just quickly is that you can see the 2 genera in blue are 2 genera from the phylum Bacteroidetes, specifically Bacteroides and Fishicola, which were clearly the most abundant in the lion gut. And before we go into those, I'd also just like to point out that there was quite a significant abundance of unclassified reads in the lion gut, which we'll go through afterwards. But first, we decided to look at some literature on Bacteroides and Fishicola and to see if it showed up in any other carnivores. And what we did find was some literature that showed it is in quite a high abundance in black-backed jackals and raccoon dogs, among others. A few other carnivores included. I see people are excited by the raccoon dogs, me too. But not all carnivores, in fact, had a high abundance of Bacteroides in their gut. For example, cheetahs actually had quite a low abundance of Bacteroides. And so our current hypothesis is that this might have something to do with the social structure of the animals. Obviously, the top 2 examples being more pack animals, pack hunting and so on, and the lower one being a solitary animal, although this requires much further research, obviously, to confirm. And then next for the short reads was the assembly and binning, so generation of our MAGs. And so what we managed to do was create 272 MAGs, I think it was. And you can see here how many we classified to the genus level and to the species level. So you can see that the vast majority of them we were able to classify to the genus level, but about 2/3 of them we weren't actually able to classify to the species level, hopefully indicating that these mags that we generated are something new and can represent some new bacterial species or other species in databases. Here we have a phylogenetic tree of the mags that we created. The phyla shown in the colors around the side, and the blue dots indicating those that were classified to the species level. So again, we hope that some of these ones on their own that are similar distances apart, but without the blue dots, might represent some new species. And here's just a more in-depth overview of what the mags looked like. You can see that our top 3 mags were Collinsella, Fusobacterium, and once again, Fischikola. Right. More recently, we've acquired our long-read sequencing, and so this is still under analysis. And you can see some parts of the pipeline here that we've done. So we've changed a bit of the pre-processing. The classification, we are not sure yet, and I'll be going through the orange section, the assembly and binning. And right now, we're still using the assembly tools, and so I'll be showing the output of Opera MS. And so here again, you can see the tree from the short-read mags, and here is the phylogenetic tree for the long-read mags, and you can see they differ quite significantly. Even though we generated a similar number of mags, in purple and pink are actually viral mags. So we had a lot of viral mags come out of the long reads, out of Opera MS at least, which is interesting. And I'll go into that a bit more here. You can see that, yeah, we have a lot of, in pink and the red, a lot of viral mags and actually very few of the others. But what we're hoping is that we can take a look at the other outputs, the other assemblies like Fly and Unicycler, and also take a look, obviously, after binning to see how this changes. And so that brings me to what I was showing earlier about the unclassified mags, the unclassified reads. Our final hope is that once we generate all these mags from the long reads and the short reads, is that we can add them to a database and use those to classify the short reads and hopefully increase that unclassified amount and see if some of our unclassified reads line up to our mags, maybe indicating again that those mags are new species. So just generally the conclusions, as we saw, Bacteroides and Fischikola dominated the line gut microbiome. A large proportion of the line gut remains unclassified, and we hope to solve this through our mags created through the short reads and the long reads. And then not shown here, relating to those other objectives, we found no significant changes related to sex or season of collection, and we were able to functionally annotate our mags to identify some common pathways in the line gut microbiome. So as I've been saying, we still have some remaining questions to answer, and what do the unclassified reads represent, of course. We have a few other assembly tools to use and a little bit to go with the long read pipeline, and also some investigation to be done and maybe some future research on why Bacteroidetes species were so abundant in the carnivore gut. I'd like to thank my supervisor, Scott, and my other supervisor, Robin, the SAM-RC for our long read sequencing, and everyone who's helped along the way. Great.
Thank you. Practical question. So I know you obviously collected the spur, and you don't want to get out the car with any of us. So how did you know it was 20 different lines?
The lines are darted, so they're asleep when we get the samples.
So it wasn't from...
No, it's not off the ground. I went in. Yes. Yeah.
Okay.
And they also, a lot of them have radio collars on, and
So you can track that,
yeah.
so you knew it was 20.
Yes.
Any questions?
So still very practical question. After they are darted, they are asleep, do you extract, how do you extract the gut microbiome? And did you try to correlate with the analysis of the microbiome profile in the feces?
So to answer the question on the collection, not to be crude, but I put gloves on, and then and then we kept them in those tubes that preserve the bacteria and froze them in Namibia until all the samples were collected, and then I went back and extracted the DNA. Could you repeat your second question on the correlation?
Correlation with fecal samples. Did you imagine initially in designing the project that you would go for fecal samples?
Oh, I see. Yes. Well, we yeah, basically yes. We planned it around the fecal samples. Yeah.
Okay, I have one more question, but in the meantime, I will get Louisa set up. So was whole genome sequencing for long read and short read?
Yeah.
So, I mean, you only really reported the bacteria and some viruses. Do you see things like, you know, there's unclassified potentially food sort of stuff, lion tissue?
Yeah.
You know, there must be some other thing, and fungal, you know, there must be some fungal components of the microbiome. Have you not explored any of those?
So first of all, part of the pre-processing was removal of host reads. So we did remove lion reads, so we're quite sure that those aren't in the samples. But actually using Kraken, we did get some archaeal reads, some from Lepidoptera, which is like butterflies and moths. I think we did get some fungal reads. So yes, we have looked into other phyla and kingdoms, but there were still a large number of unclassified reads, even when considering we used the large Kraken database that has plants and yeah, and there were still some unclassified reads. Yeah.
Thank you. Thanks again to the speaker.